o'clock, I think. Very good. Okay, so good afternoon, everybody. It's really exciting to be here for this seminar, the first seminar of the academic year 2024-2025. Sorry, 2025-2026. It's we cannot just take it for granted. No, it's not exactly. We cannot take it for granted that we are here. So thank you, all of you, for being in person. Thank you to all of those of you who are joining remotely. For those of you who don't know me, maybe some people online don't know me. I am Ana Navas. I am the director of the Columbia University Northern Plains Superfund Research Program. And we are really excited. This Superfund Research Center brings together people, investigators and scientists here at Columbia University, together with our partnering communities and research, local research organizations in the Northern Plains, especially our partners from Missouri Breaks, as well as other communities. As people are joining in the room, I can imagine that the same is happening in Zoom. People are just popping in Zoom. So it's always good for me to start a little bit slowly so that we have time for everybody to be ready. And for this first seminar of the academic year, we are going to have a special discussion and topic, which was originally brought to us by our community partners in the Northern Plains. Manganese is a very interesting trace element. Is an essential element that we need for our functioning, but it can also be toxic. And because of this, there is concern about it. There is some interest to regulate it. There are some levels that are considered potentially toxic, although the regulation is not totally finalized at the same time. And for that reason, it's an element that is very difficult to study in many ways. And this is true in part because there are not good biomarkers also of exposure in humans. So today we are going to do two things that are really interesting, I believe, I hope so, for everybody. First, we are going to learn from the communities that have been affected by manganese exposure. They are telling us what happened and why they want to, and how we could help also, how we could help with the data that we have collected as part of the Superfund Research Program. And then we are going to go into a presentation, a scientific presentation, to learn more about manganese metabolism, and the interactions in the human body. So with that, I think we can invite Vicki Alberts to share with us more about what happens in the Northern Plains, in particularly in the community, the Spirit Lake Tribe community.
All right. Well, thank you again to everyone for allowing me to take a few minutes of your time, and especially to Ms. Ray O'Leary. I really appreciate the connection that we've developed and the ability to coordinate with Columbia, and we definitely appreciate all of the work that has been done through the Strong Heart study here at Spirit Lake. Just a quick introduction of myself. My name is Vicki Alberts, and I am currently the public relations specialist along with a law clerk here at the Spirit Lake Tribe. As you can kind of tell from my title, people are probably wondering why I'm talking about the elevated levels of manganese. Well, I will tell you one thing: we definitely wear a lot of different hats within the tribal realm, and mine just happened to also fall into the hat of helping to coordinate the efforts with the elevated levels of manganese that we experienced starting in May of this year. And as Ray is kindly sharing her screen, we have an incident timeline ranging from, we dealt with this issue starting May 13th, and finally were able to officially lift everything and kind of get back to a normal status on August 20th of 2025. So there was about a 3-month period where our residents, about 5,000 residents within tribal boundaries, were not able to consume their drinking water. And everybody who resides within reservation boundaries is all on the same water system. And our water level in the elevated levels of manganese were detected in mid-May, about May 13th, 2025. And as a result of that, we began to notify, when it came up on our radar on May 27th, 2025, we were already at the point where the elevated levels of manganese were about 1.23 milligrams per liter in the drinking water. And as we know, the EPA standard, that's well above the EPA standard. So we had to act quickly, because we have elders in our community, we have young infants. And so there was obviously a lot of questions. How did this happen? Where did this start? How did this begin? And also, what do we do in this situation? And here at Spirit Lake, our tribe has learned to utilize the resources that we have in the valuable assets of our people who work for the tribe. And they created we developed an incident command team. And as a part of that incident command team, this was our first kind of go around with a large event, or kind of a large scale event that affected all the residents here within reservation boundaries. And immediately we had to find drinking water, ensure that we were getting proper drinking water to our community members. In addition to just having locations designated for our water efforts, we also had to take into account, we live in rural North Dakota, and I'm sure many of you probably don't wish our winters on your worst enemy, where it can get to -50, with wind chill, and you don't know it until you experience it. And so thankfully this happened in the summer and not in the -50, below temperatures. However, the fact that it happened at all was a bit of a detriment to our community. However, like I said, our people came together, and we were able to coordinate efforts and get drinking water to our community members. And that included taking into account people who had no transportation. We are a part of a tribe, one of the 575 federally recognized tribes here in the United States. And unfortunately, that is a reality, is that not everybody has transportation. Many of you and correct me if I'm wrong, but many of you live in more urban settings, much more urban than, where we are. And so there might be public transportation, easy ways to kind of get around without a vehicle. However, here we have miles between homes, and people who have, severe medical needs that they're not able to get out and pick up water. So we had to create not only an entire inventory system of water distribution, but we also had to create a delivery system for those individuals who weren't able to make it to their community centers. But we distributed water to our community starting in May, the day of the incident, all the way up until about August 15th is when we started pulling away. However, we were keeping track as much as we could of these levels, and to see where the water levels kind of started to go down a little bit with the work that we were doing. And then in the middle of it, in July, we moved over from our original system to a temporary water filtration system that was of limited capacity. So we were pushing out on our normal operating system about 1 million gallons per day on our normal filtration system. And when we had to operate on this temporary system in mid-July, then we were operating at a rate of about 750,000 gallons per day. So we had to move from a water, ban, not drinking, to a water conservation and consumption ban. So we were doing two of those things at once, until we were finally able to get the water levels down. And as you can see, on July 2nd and 3rd, we were able to demonstrate that our water level was starting to finally go down with the proper filtration system. But a part of that for us was not only dealing with fixing our large scale filtration system in our water supply system. It was also the fact that the media that we needed to do that was unavailable until August. So it was a good 3-month wait. And so our people were very patient, and that was the biggest thing for us, was to ensure that the health and safety of our tribal residents was top priority, and ensuring that they had proper drinking water during that time. And if you look on I have a full scale timeline, which I shared with Ray. And if anybody else is interested in taking a look at this, I mean, you're more than welcome to do so. But on July 9th was when we were officially able to lift our water conservation ban and finally allow people to get back to somewhat normalcy with drinking water. But we still had that not the consumption ban, but the what do you call it, conservation in place until we were able to get our full operating system back on par. And that again didn't take place until August 20th. So it was an entire summer. Our children ended school with this, and our children went back to school with this issue. However, it was definitely a learning process, and we are forever grateful to the Strong Heart Study, because the organization also played a major role in helping us to look at some old data from years back, looking at manganese levels to kind of determine and give us an idea as to perhaps when did this occur? Has this been an ongoing issue for years? And we were just lucky enough to have that connection with the study to review previous years of water testing and water levels. And we were able to kind of pinpoint almost a starting point, for this issue. However, it was something that we don't wish on any other community, yet it was definitely, it was a learning experience. And we had to ensure that we had, safe drinking water for animals, for our residents, we had large community events happening at the time. And so making sure that our visitors also had access to good, clean water in these larger events. But it was a pretty large scale crisis, I guess, is the best way that I can put it. Yet thankfully, we are back online with our normal water limits, but we are very grateful for the Strong Heart Study and their data that they were able to provide to the tribe, along with an explanation to kind of help us determine, maybe has this been going on for years, or is it something of more recent start. But I don't know if there's anything specific that anyone would like me to address. I'm more than able to talk about it, but that's just kind of a real brief synopsis of what took place here at the Spirit Lake Tribe in Fort Taunton, North Dakota.
Vicki, thank you so much for sharing this experience. I think it's really eye-opening for all of us here. Also, I would say that these are your data. The data that we gave you back, in a way, was your data. And we were very happy to be able to make it useful and usable for you. I want to thank everybody in the team who really worked really hard to make the data as easy as possible to interpret for you, preparing figures, graphs, making the database simple enough so that you could really use it in hopefully easy ways. So thank you. Many of you are here in the room and probably online also. So with this, I am afraid of taking minutes for questions, because I think we need to keep moving. But we are going to learn now much more about manganese. What is it? As I mentioned earlier, it's a complicated element. And to enlighten us, we have today Dr. Rebecca Haeusler, Vicki Hauesler. And Vicki is an associate professor of pathology and cell biology. She also has a joint appointment in medicine or in medical sciences, I believe. And in terms of training, she did her bachelor at MIT and her PhD in biological chemistry at the University of Michigan. And we've had the pleasure to learn a lot from Vicki in the last few years, and all the great work she's doing on manganese and metabolism and liver specifically. So thank you so much, Vicki, for being with us.
Okay, thank you so much for inviting me here. Really different audience than I'm normally speaking to. So that's going to come up while we're having this talk. I did want to dwell for another second on where my appointments are, because I think it will help you understand my take on things and where I'm coming from and what I know a lot about. So my lab is here at Columbia in the Diabetes Center, across Broadway and St. Nicholas. And I'm also part of the leadership team for our Digestive and Liver Disease Center and our Diabetes Center. My primary appointment is actually in medicine as well as in pathology and cell biology. And my research lab is really focused on understanding insulin action and how it controls metabolism. So we think a lot about glucose, triglycerides, cholesterol catabolites like bile acids, and then how those things go wrong in diseases and conditions like insulin resistance and obesity. And so, again, coming back to this point, like this is a really different audience, and we all have different expertise in this room and on the call. So you can stop me at any time, ask me anything. There's no dumb question here, and you can let me know if anyone has a question on the chat, because really we can. I didn't make so many slides. We should have time to stop and talk about things if there's anything that's not understood. Okay, so this is another slide to explain the background of the project. I'm going to have just a couple of background slides about our lab and what we were studying that brought us into manganese. And so we're, again, interested in insulin action and how it controls metabolism. And one of the key sites for insulin to act is on the liver. Okay, that's one of the primary sites where insulin can act. The liver sees higher levels of insulin than any other tissue because of where it is next to the portal vein. It's getting all that insulin directly from the pancreas. And so there's a lot of things that the liver is known to do in response to insulin. Okay, so this is a depiction of the hepatocytes, the primary, the major cell type of the liver. And inside of the hepatocytes, when insulin signaling occurs, it regulates a number of different processes that we understand from like decades of research, that insulin signaling in hepatocytes regulates how much glucose is put out from the hepatocytes into the blood, how much lipid is taken up or oxidized or new lipid is made, and the secretion and uptake of lipoproteins like HDL and LDL, and the secretion of other secreted factors, other hormone-like proteins that are secreted from the hepatocytes. And these are so regulated by insulin signaling and also can be disrupted when a person becomes resistant to insulin or gets type 2 diabetes. And these are really important ways that the liver can communicate with the entire rest of the body, because these are ways that the liver mediates the secretion and uptake of a huge variety of macromolecules into and out of the bloodstream. So it's talking to all these different tissues. And what I want to point out here, and where our sort of unique, and these are areas, by the way, that we've worked in a lot, but where our sort of unique contribution lies is in the fact that the hepatocyte is actually what we would call a polarized cell. It has a membrane facing the blood, and it has this totally separate membrane that faces a totally different direction, which is towards the bile. So I've labeled the bile canaliculus. It feeds into the bile ducts, which of course would travel down into your intestine. And the hepatobiliary system mediates the excretion of a lot of different important molecules, including some metals, and cholesterol and other things, and can therefore mediate not just excretion, but also crosstalk to the intestine and signaling processes. But I put a question mark here because the impact at this side of the cell in conditions like insulin resistance or diabetes is a lot less understood, just like not very well explored. And so that's where our expertise comes in. And so I just pointed out on the other side of the slide here that after a meal, your gallbladder contracts, allowing your bile to pass down through your bile duct into your intestine. And then a lot of the biliary constituents are actually reabsorbed. So they undergo this enterohepatic circulation, including, most of the cholesterol is reabsorbed, the bile acid, the manganese, et cetera. Okay, so we got into this this side of the cell through our long-standing interest in the conversion of cholesterol, the breakdown of cholesterol into bile acids, which are the primary breakdown product of cholesterol. And so what we had been working on over many years is the fact that insulin signaling controls this process, controls the production of bile acids and which bile acids get made, and how well those bile acids are transported out from the hepatocytes into the bile. So this is our area of expertise. And then what I'm going to tell you today is this manganese story that sort of sprung from those studies and ended up being one of those kind of projects where you're simply following the science where it is leading you to something that was really unexpected, but turned out to be really interesting and actually brings us back to insulin signaling in a totally unexpected way. So this slide is going to be a single slide that is the background of the manganese project, why we started working on manganese. So again, we were working on how insulin signaling, the fact that it regulates the types of bile acids that get made, and the fact that that is then altered during insulin resistance. So is that meaningful? Does it have any consequence that insulin would affect or insulin resistance would affect the composition of the bile acids? So a former PhD student of mine that, Anna, you actually met her, she presented in our manganese or our metals and health seminar series once a long time ago. So she wanted to ask the question, what are the consequences of altered bile acid composition in the intestine, the terminal ileum, the end of the small intestine, which is a site of very high concentrations of bile acids. A lot of bile acid signaling happens here. So in this place, what does it mean to have different kinds of bile acids? So for this, she used a model of the intestine, which are called enteroids or organoids. You can use intestinal stem cells, differentiate them into a dish to make miniature intestines in a dish. And then she treated them with designer bile acid pools that would mimic the extremes of insulin sensitivity versus insulin resistance. And she did this for bile acid pools that would be like people or like mice, because there are some differences between mice and humans, and we wanted to see what would be overlapping. And so then she submitted the organoids for RNA sequencing, and we saw like thousands of gene expression differences just by the types of the total amount of bile acids is the same, just the type of bile acids that's being treated on these organoids. So then how are we going to figure out what we want to follow up on and really like dive in deep and learn something here? So we decided to pursue genes, or at least a gene that would be very reproducibly induced by bile acid, that would be the directionality we're thinking about, but differentially induced by high versus low, these differences in the bile acid pool, but conserved between mice and humans, because of course we want to do work that's the most translational possible, and extensively validated by ourselves and others. And the gene that she ended up focusing on was this gene, SLC3810. Which turns out to encode this manganese efflux transporter. And what Tiara figured out for her dissertation and her paper was that bile acids induce this transporter by acting on the vitamin D receptor, which is a transcription factor, and that this has the functional consequence of causing manganese efflux out of the intestine into the lumen of the intestine. So this seemed really interesting and rich, and I am now going to introduce you to the fundamentals of these transporters and how manganese levels are maintained in the body, and why it is of interest to us, given our background in metabolism and insulin signaling and the diabetes center. So manganese, as you know, is an essential micronutrient. We have to consume it. We have to get it from the diet, and it would be, of course, absorbed from the intestine, and then absorbed into the bloodstream. And excess manganese has to be excreted through the hepatobiliary system. So it's going to be excreted into the bile through that process that I described to you, and then released back again into the intestine. And so if we zoom in on the cells mediating this process, these would be the two cell types that mediate the manganese concentrations in our body: the enterocytes, the absorptive cells of the intestine, and the hepatocytes of the liver. And each of those cells is a polarized cell. So the enterocytes have a side, facing the lumen of the intestine, and the side facing the blood, and the hepatocytes, just as I described before. And on these different membranes, there are a handful of known transporters that are known to be involved, known to mediate the uptake of manganese from the environment into the cell. And you don't have to remember the names of these. I just want to point out that the identity of these is pretty well established. We know how manganese, by and large, is getting into cells through these uptake transporters. And yet there's only one known efflux transporter for manganese, which is this one, SLC3810. So it is expressed, it is localized on the canalicular, the bile-facing membrane of the hepatocytes, only on this side, and on the luminal side of the enterocytes. So it can take manganese from the inside of the cell to the bile, or from the inside of this cell out into the lumen. So in both cases, trying to get manganese out of the body. And the discovery of this transporter was in 2012 by two groups that had been doing the genetics of hypermanganesemia. Okay.
What kind of proteins are they?
They are solute carrier transporters. Yeah, so they use a gradient. Some people believe it's using calcium as going in the opposite direction as manganese. Yeah, does that answer your question? Yes. Okay, so why would we be interested in continuing this research given our interests? And the reason is because of these old studies showing that manganese is known to affect glucose metabolism in people. So this was a study from 1962 published in the Lancet by Arthur Rubinstein. I don't know if you know him, very famous endocrinologist. He was the dean at Penn, president of the American Diabetes, very famous. And when he was still in South Africa, he made this case report of an individual in South Africa who was self-medicating his diabetes with a local folk remedy that was an alfalfa extract that they determined had a lot of manganese in it. And they could replicate the glucose-lowering properties of his alfalfa extract using manganese chloride alone. So that was one individual, but another totally independent research group then followed up on that with a much more rigorous study, and they looked for individuals who had manganism, so they had known, confirmed high manganese levels, and in this case, it was from exposure from a local mine in Egypt. And then they matched those individuals. They had 11 male subjects who had manganism, and then they matched those to age, sex, pretty well matched controls, and they had 20 of those, and they did an inpatient study. These were all inpatient. They fed them a high carbohydrate diet for 1 week, and then they did this intravenous glucose tolerance test. So they gave them a glucose bolus intravenously, and then this is what they saw. People with high manganese had lower glucose levels, blood glucose levels, throughout this glucose tolerance test. So manganese had glucose lowering properties in these studies. And these studies were published, even this follow-up was, more than 50 years ago. How could this all be happening? Well, that's pretty hard to determine if you can't tightly control manganese. As you said, it can be kind of hard to study. And it can be especially hard to study if you don't know how to tightly manipulate it. And so now we can do that because we have these genetic tools, because we know the identity of these transporters. So we can use genetics to understand the effects of manganese on metabolism. And for our case, we are particularly interested in liver because, again, it's a hub for manganese handling and transport. It's used to handling all the manganese that needs to be excreted. High levels, higher concentrations of manganese in the normal state are here than any other tissue. And it's the major hub for glucose homeostasis. So if you were going to guess how those two things were brought together, you might think that it would be happening in the liver. So this is the way that we used mouse genetics to test the metabolic effects of manganese in liver. Okay, so we obtained, and this was done by a PhD student in my lab that many of you probably met. She attended many of our Metals in Health, although she just defended. Maybe she's online, I'm not sure. Jen, she is now a postdoc at Penn. Okay, so we obtained these mice that had floxed alleles for this transporter so that we could knock it out in a tissue-specific conditional way. And it turns out that this thing is expressed, as I said, on the hepatocytes and the enterocytes. If you only knock it out in one or the other, essentially nothing happens because the other one can compensate. So if you only knock it out from the hepatocytes, the excretion of manganese can just be rerouted and be almost entirely then excreted from the blood across the intestine into the lumen of the intestine directly without engaging the biliary system. So you have to knock it out in both places if you want to increase the manganese in the body. And I guess that says something about how much we don't want to have manganese accumulating in our cells. Okay, so the way that we approached this was to take the mice, we crossed these floxed mice with a villain Cree that will knock out the efflux transporter from the intestine. And then the mice there's nothing wrong, there's not manganese accumulating, they go through development totally normally. And then after they're developed, through weaning, then we give them an injectable Cree that deletes the SLC3810 from the hepatocytes. And then we wait a couple of weeks and it has the desired effect. So now we're using the metal lab, the core facility here, ICP-MS, measuring the liver metal content. You could see that just the 3 weeks of knocking out the transporter in both places has the effect of raising the manganese content in the liver. The concentration is higher, but it does not have effects on other metals as you would expect because it's specific for manganese. And I just put a couple of these here that are just to remind us that number one, other metals are not changing, and also that the concentration of manganese is naturally much lower than many of the other metals. And so the y-axis is totally different here. And this is going to come into play later when we're going to do some comparisons between manganese and magnesium. Now, you're surely aware of studies where high levels of manganese, maybe from exposure, but also from human genetics, chronic lifelong loss of this transporter, or various SNPs, and we can come back and talk about these more, are associated with liver injury. So is that happening in this model, in this system of what we're doing here? And that's not happening. So AST and ALT are two blood markers of liver injury, which are completely normal in these mice. So we're not looking at a model of liver injury in this setting. These mice are just on a regular chow diet. They also have totally normal body weights, but what they have is a reduction in their fasting blood glucose levels. And that occurs without any change in the insulin level. So, these are normally very tightly homeostatically connected, but they do not have lower glucose because they have a higher insulin, for example. If you give these mice a glucose tolerance test, in this case, I'm showing you an oral glucose tolerance test, they, like the humans, have lower glucose, blood glucose levels throughout the glucose tolerance test. So they have a glucose lowering effect, these mice with higher manganese. I'm not going to show you all of the data, but this occurs in female and male mice. It occurs whether you give glucose orally or intraperitoneally. We challenge the mice to a more obesogenic diet, high fat, high sucrose, and they are protected from the hyperglycemia in that setting. But there's no change in insulin secretion. If you give them oral glucose, they have the same insulin secretion, actually slightly lower insulin secretion. Again, the glucose is not lower because they secreted more insulin. There's also no change in what we call insulin tolerance. If you inject the mice with insulin, it causes the muscle and fat to take up a lot of glucose, and that occurs totally normal. So muscle and fat seem to be fine. But what is different is that if you give the mice a bolus of pyruvate or glycerol, which are precursors for producing glucose in liver, they have a reduction in the glucose excursion. So we're looking at a liver effect. So all of these together closely mimic many previous examples of genetic or pharmacologic tools that increase hepatic insulin action. So this is a model that mimics increased hepatic insulin action. One thing that we did to kind of go along with these data is to explore other aspects of hepatic insulin action, because it's not that the only thing it does is lower glucose in the liver. And so we looked at a couple of other markers of increased insulin action in liver, which some of them, I guess, may surprise you. But insulin, the normal action of insulin, is known to potently stimulate the synthesis of lipids. And if you have mice with higher insulin action, a higher insulin sensitivity, they will develop a fatty liver. And that does happen in these mice. So they have improved or lower glucose, but they have more liver fat. Insulin also, I told you we have this longstanding love for the bile acid pathway. And one of the things that insulin does is change the types of bile acids that get made. And this is this model is completely mimicking everything else we know about insulin regulation of bile acids. So I put these in here to say that these effects may be relevant to the discussion at the end, that it's not just glucose lowering that hepatic manganese is doing. Okay, so then let's talk about these fundamentals of insulin action. Insulin, of course, is secreted from the pancreatic beta cells, and then it lowers blood glucose by acting on the muscle and fat to promote glucose clearance from the blood. And then it also acts, as I said, on liver to suppress the production of new glucose. And this occurs via a very well-known, very well-established signaling cascade. So proteins that are modulating each other to enact these effects. And so let's look at the insulin signaling cascade. So it starts by insulin binding to the insulin receptor at the cell surface, and then there is a phosphorylation cascade. These proteins end up becoming phosphorylated to become activated. And you don't have to remember any of these except this one. The protein, the key protein that's in the middle of this cascade is this protein, AKT. It's a kinase. So its role is to phosphorylate other proteins. So like this is the central mediator, and it goes on to activate all the other targets of insulin signaling inside the cell. So that's how the signal is amplified across a lot of different pathways. And so some of the really well-established targets for AKT in hepatocytes, these are phosphorylation events that AKT carries out, and those have the consequence of then raising the synthesis of glycogen and lipids and suppressing the production of glucose. But insulin and AKT are acting in a lot of different cells, and AKT is a very famous protein, and it's acting in all those different cell types to mediate other pathways of metabolism, growth, proliferation, and survival. Now we're going to go a step deeper on this protein, AKT. AKT has these various domains of the protein that cause it to be in a particular shape, a particular conformation when it's inactive, which would be like when insulin is not signaling. And the key thing here is that it has this lipid binding domain that is covering the catalytic, the functional domain. So like it can't do anything because it's physically blocked. And then when insulin signals, the way that it activates AKT is twofold. Number one, actually threefold. This upstream protein phosphorylates AKT. A second protein phosphorylates AKT on another site. And then this lipid binding domain flips up and binds to the membrane. So now we have two new phosphorylation sites at threonine 308 and serine 473. And this lipid binding domain is binding up here. So now it's made a huge conformational change so that it becomes an active form of the protein. And this allows the protein to be like open for business, and its targets, like this protein GSK3 beta, we're going to see a lot of information about this protein, can come in to the catalytic domain and become phosphorylated by AKT. Decades of research indicates that what I just described to you, membrane localization and the phosphorylation of threonine 308 and serine 473 are the primary activators of AKT in response to insulin. So let's get back to our mouse model and how I showed you that when we increase manganese genetically, it mimics increased hepatic insulin action. So we want to test that directly, and we can do that in mice by taking our double knockout mice that have the higher manganese in liver and injecting them with insulin. We inject it in the jugular vein, and then 15 minutes later, we look at the insulin signaling cascade in the liver. Now, you don't have to look at all of these Western blots. I just want to point out that here we've loaded on this gel. Every other lane is a knockout mouse, so every other lane has the higher manganese. And what you can see are really big differences down here. All the proteins that are downstream that are intended to be phosphorylated by AKT are phosphorylated better, or more so, more robustly in the mice with higher manganese. And these are just like some individual examples, but we can also use, it's a little bit easier to see on this sort of smear where we can detect using an antibody all the targets of AKT. It gives us this sort of smear of like a huge variety of different proteins that are phosphorylated by AKT. And you can see in the knockouts, they're phosphorylated more robustly in the mice with high liver manganese. At the same time, this is not happening to all proteins or even all kinases, and there is no change in the liver activity of a very highly related kinase, which is called protein kinase A, PKA. And also, it seems to be specific to liver because there's no change in AKT activity in the soleus muscle or the epididymal white adipose tissue. So we have a liver-specific increase in AKT signaling. So we want to ask, is this therefore an effect directly of that manganese, specifically on the hepatocytes in a cell autonomous way? You know, when you're looking in vivo in a whole organism, you can't sort out that the manganese acted on the pancreas to then affect the liver. But if we isolate the cell, so here we're isolating the primary mouse hepatocytes, and we treat them with a cocktail forskolin and dexamethasone that mimics fasting. Okay, and then we can treat them with insulin and manganese to see what happens. And what we can observe is all, you know, if we look here just at this insulin treated lane, all the expected effects of insulin occur. So we're turning on AKT phosphorylation, what I described to you at these two key canonical sites. And we're also activating the phosphorylation of the downstream targets of AKT, like GSK or like in our smear. If we look at the cells treated with only manganese or with both of them, what you can see is that manganese alone is sufficient to turn on the targets of AKT. The AKT phosphorylation is activated with manganese alone. And I want to point out that that is occurring without apparently, at least in these cells, any appreciable phosphorylation of AKT itself. And that's going to become important later, which is expected to be the key way, the textbook way that AKT is activated. And also, this is, again, just like we saw in livers, now in these hepatocytes, again, there's no change in the activity of this closely related kinase PKA. And I just want to put on one slide to say, you know, we're using mice as a model, but we care very much about what's happening in people. That's why, you know, I started with those human examples, but we also use primary human hepatocytes that we can source from a distributor who collects, you know, livers from unfortunately deceased individuals and makes primary cells. So these are human primary hepatocytes, and we see the same thing. Insulin turns on the cascade or manganese, or the combination. And we repeated this from multiple independent donors. Yeah. I'm going to touch on that later. It is more. We did that quantitatively. We were very curious about that. And I have a whole slide about that, or two slides. So those are all, you know, Western blots, like signaling cascades, okay, very like cerebral, but like, is it having a functional consequence that mimics what's happening in vivo? Is there a functional consequence on the production of glucose in those cells? So you can, you know, you isolate the primary hepatocytes and they're producing glucose. That's what they do. And you can suppress that with insulin, or you can suppress it with manganese. And so they are having this functional consequence on glucose, and that is going along with the expected changes in gene expression that are predicted, that are, you know, well established to mediate those effects on glucose production. And it goes along with a lot of other gene expression changes that occur in response to insulin. You know, all these gene expression changes that are shared between what insulin does and what manganese does. And then, of course, there's things that are different between, you know, only insulin does or only manganese does. Importantly, we also did these dose curves. So in cell culture media, there actually is no supplementation of manganese. Interestingly, even though manganese is an essential element, and your media is supplemented with zinc and magnesium and calcium, not manganese. So you're getting like random amounts in your various batches, and so but they're low. And so we supplemented the cells with low doses, and you can see this dose-dependent response, which is in the physiological concentrations of blood manganese. Okay, so what I just showed you is that, yeah, yes.
I have a question.
Yeah.
We're showing statistical differences. Top.
Yeah.
To the bottom.
Yeah.
But isn't the manganese only different from the insulin only, and also different from some of those manganese? I mean, it's small, but it looks quite a bit higher.
You know, it could be that if we had a higher N number, you know, this is like a lot of different conditions to have enough cells from the mice to do that. But, you know, it could be just insufficient N to see that difference that you're like observing. Yeah. If we had a lot higher N, that's possible. But also, you know, it could be, you know, We put here 6 micromolar of manganese. We put 1 nanomolar of, all those that could be different based on the doses that we use. Yeah. So what I just showed you is that in vivo in mice, ex vivo in primary cells, and in some cell lines that I didn't show you, manganese is sufficient to increase AKT activity and suppress hepatocyte glucose production. So for us, a key question is like, where and how is manganese acting to do this? So let's revisit the insulin signaling cascade and how AKT is regulated. We know that the effective manganese requires AKT because an AKT inhibitor blocks the effect. Now manganese cannot turn on these targets. Okay, great. So it really is AKT, but it does not require signaling upstream of AKT. We use an insulin receptor knockout model. We have no insulin signaling. Now insulin can no longer turn on this pathway at all, but manganese still can. And I already showed you that AKT phosphorylation, which is the canonical way that insulin turns on the pathway, is apparently not required, because when you treat cells with just manganese, this is not becoming phosphorylated. So it led to the hypothesis that manganese could be acting directly on AKT protein. So what is the evidence that manganese could be acting directly on a protein like this kinase AKT? To understand that, let's do a deep dive into the basics of a kinase. What does a kinase need? This is a protein that is trying to donate a phosphate group onto a substrate protein. So it needs the catalytic domain of the protein. It needs the substrate that it's trying to phosphorylate. It needs ATP that's going to donate the phosphate. And then what it needs to make the reaction go is a divalent cation, 2 of them. And it is widely believed, it has always been believed, that kinases in vivo, in cells or in an organism, it has been presumed that they are relying on magnesium. And that's an understandable assumption, because magnesium is very highly abundant, orders of magnitude more abundant in liver than manganese. And because for the model kinases that have been studied for many decades, like PKA, magnesium works best, better than other metals. But it is also known from in vitro studies where, using a cell-free assay, using purified kinase proteins, that other metals are capable of supporting kinase activity in that setting, not just magnesium, but also cobalt, cadmium, and manganese. And here is where I want to point out that even though something like 1/3 of our proteins bind to or require a metal, the metalloproteome is not solved. It's not known in vivo which proteins are using and binding to which metals. This is like a really hard problem to solve. And so here is some direct evidence. You can directly visualize the possibility that manganese is acting directly on AKT. And this comes from crystal structures of AKT, of which there are numerous. So what I'm going to show you in the green trace, this is AKT protein crystallized. So they've determined the structure of this protein that's actually lacking that lipid binding domain. And then in the purple trace here is the substrate protein, GSK. And then back here in the orange is the ATP. It's actually an ATP analog called AMP-PNP. If you put ATP, it would just, like, the reaction would just go, and then you wouldn't have anything to crystallize. So they put in this analog that cannot be hydrolyzed. And then if you look back here, these 2 purple dots, I'm going to zoom in on them. Those are 2 manganese ions directly bound in the catalytic domain of AKT. And they're coordinated by the 2 highly conserved metal binding residues that are present in all AGC family. The entire family of kinases have these exact 2 residues. It's just that they were thought to use magnesium. So manganese is present here. So now let's do our own experiments to test this functional effect. So we used here, these are cell-free, not in mice, not in cells, in a tube, purified AKT protein. Here we're using the full-length protein, and we can see that just adding manganese is sufficient. You add the ATP and the substrate. If you just add manganese, if you don't have any divalent cation, the reaction cannot go. If you add manganese, it is sufficient to induce the activity such that AKT can phosphorylate its target, GSK. Then we look at the other activity of a kinase, of AKT, which is that it needs to hydrolyze that ATP. It needs to get the phosphate off. And so if we give it manganese alone here, it is sufficient to activate the ATP hydrolysis, and it works better than magnesium, even at much higher doses. And so if we directly compare how well manganese versus magnesium activate AKT, what you can see is that we have this very clear and potent effect that manganese increases what you will call the catalytic efficiency, how well the reaction turns over. And I want to contrast this directly against that other kinase that we've been bringing up, PKA, which has been, again, a model kinase. Many people have studied it and have established that it works best with magnesium, and that's why we made all these assumptions about all the kinases. And PKA really does work better with magnesium for some reason, even though they're very similar. But that's not true for AKT. Finally, AKT is, as I told you, has this full-length version that has this lipid binding domain that normally keeps the protein inactive. And so if you want the protein to be like more active, even in vitro, you have to cut that part off because you can't bind it to a membrane in vitro. And so you cut off the lipid binding domain. And now what we're seeing is that manganese turns on this AKT, the catalytic activity of AKT, even at these micromolar, physiologically relevant micromolar doses of manganese. So what is the relationship between AKT activation by insulin, which is very well established, versus manganese? Are they completely independent or do they work together? We were very interested in this. And so the way that we addressed this, we wanted to have something more quantitative than our Western blots. And so we used a phosphoproteomic approach. So we took our primary hepatocyte model, we treated them with, the forced dexamethasone, mimic fasting, treat with insulin or manganese. And then we submitted those for total proteomics and phosphoproteomics so that we could quantify the signaling cascade. And what we saw is that, as you would expect, insulin induces 300 phosphosites in hepatocytes. That's just what you would predict. And then we broke them down into the categories of how manganese acted on those. So there was only a minority of the phosphosites that were unaffected by manganese or going in the wrong direction. The majority of them were also induced by manganese alone. And then we used a biostatistical core to help us statistically analyze whether the effects were additive. So the effect size of manganese plus insulin, was it significantly greater than manganese alone and insulin alone, and then narrowed that down to the ones that were statistically significant. And there are 70 that we consider to be significantly additive. So like, what are those additive sites? Are they something weird? Well, they're not something weird. They're like all the known canonical effects of AKT. And if you were an insulin signaling biologist, you would recognize all of these sites: TSC2, FOXO3, GSK3 beta, IRS2, ACLY, FOXK1. These are the ways that insulin acts in cells. And these phosphosites are being activated by, the labels are down here, by insulin alone, by manganese alone, and together even more so. So they are acting additively. And so then we wanted to understand what the identity of those, of these different categories of sites were. So we took a deep dive into the motifs, so the string of amino acids near the phosphosite that determines which kinase is going to phosphorylate that site. And we can use like known motifs that are known to be phosphorylated by certain kinases. So among all the insulin-induced sites, all the 300 sites, about 30% of them have a canonical AKT phosphorylation motif, an arginine, any amino acid, arginine, XX, and then the serine or threonine, or something similar. If we look at the sites where insulin and manganese act in opposite directions, that is de-enriched. And if we look at these sites where they're additive, it's enriched for sites that are AKT sites. So it seems like the way that insulin and manganese are acting together is through AKT, as we predicted. So manganese seems to act directly on AKT to stimulate its activity. The metal selectivity of AKT preferring manganese is relevant even in vivo. Is this a natural mechanism used in hepatocytes to control AKT activity? Like, is this natural in any way, or have we just made this happen by our genetics. And so a crucial setting where hepatic AKT wants to be primed for maximal activation potential is in that transition between fasting and feeding. Insulin acts within seconds to minutes, 30 seconds, 1 minute, you're maximally activating the insulin signaling cascade. So between the fasted state and the fed state, you really want AKT to be ready for that maximal activation. And so that led us to investigate that transition between fasting and feeding and the nutritional regulation of liver manganese. And here I just want to point out, because you may be thinking about your studies of blood manganese, and unfortunately it turned out for us that liver manganese was not regulated like blood manganese. It's a hurdle for us to overcome, to work together. But anyway, I just wanted to point that out. Okay, so again, we've got this efflux transporter that we care a lot about. That's going to get the manganese out of the cells, and then these other transporters. The other transporters I'm actually not even going to show you because they turned out not to be regulated by fasting and feeding at all. But 3810, the efflux transporter, is very strongly nutritionally regulated. So this is a fasting and feeding study in mice. So we took mice and we harvested tissues throughout a 24 hours, up to 24 hours of fasting at different time points, or different mice that we had refed at different time points. And what you can see is that during the fasted state, 3810, the efflux transporter is low. And then in the hours after feeding, 3810 turns on. And so the directionality, I know it's hard to wrap your head around, is that this would be the state when you're holding manganese inside the liver. And this would be the state when you're hours after feeding, you're going to be releasing your manganese into the bile. And so we took a look at the liver manganese concentrations during this refeeding setting. So indeed, at the end of the fast, this is like that 24-hour fasting time point, the liver was enriched in manganese. And then in the hours after refeeding, coincident with the induction of the efflux transporter, we see a reduction in the manganese concentrations. And so how does this occur? Not through insulin. It turns out to be occurring through carbohydrate signaling. This is, I think, my last data slide, so rest easy, we're almost done. Carbohydrate metabolites are well established to regulate gene expression through their own transcription factor, the carbohydrate responsive element binding protein. And that turns out to be the key transcription factor that is mediating the ability of carbohydrates like fructose. If you just give mice fructose, you can induce SLC3810, but not in the mice that lack CHRBP. And this is because the CHRBP binds directly to the promoter of this gene, activating its transcription. And so, again, we are really interested in functional studies. Does this have an actual consequence of just gene expression? Does it have an actual consequence on manganese homeostasis? So we did these studies using bile cannulation, where we ligate the distal common bile duct. We divert all the bile into our tube, and we can measure then the hepatobiliary excretion of the thing of interest. So here what we've done is taken wild-type mice and given them a bolus, an oral gavage of water in the black line, or fructose. We just gave them fructose, and that was sufficient 2 hours later to triple the rate of biliary manganese excretion. Well, there was no effect on copper, the other biliary excreted metal, and no effect on the bile flow rate. So this is our model. We're looking at the hepatocytes. SLC3810 is normally expressed at the canalicular membrane, but it's low in the fasting state. So it's going to allow manganese to accumulate inside of the cell, prime the cell, probably priming the ATP pool. And then after insulin signaling occurs, AKT relocalizes to the membrane, becomes phosphorylated, in this way. And also now it has a lot of manganese to rely on to rapidly carry out its desired effects. And then slowly after the meal, this occurs very slowly. Actually, the actual mediation between carbohydrate metabolism and the activation of this is still under debate, but it occurs slowly over hours. So this, over the hours after the meal, this turns on, and then turning on the efflux transporter, allowing the manganese to be released out of the cells and into the bile, lowering the concentrations in the liver. So the take-home points here are that raising manganese in hepatocytes has glucose lowering and insulin mimetic effects in mice and primary cells from mice and humans, mimicking effects in humans reported 50 years ago. Liver manganese concentrations are naturally controlled by carbohydrate metabolism in ways that we did not previously appreciate. And so we would propose that controlled access to manganese ions is an insulin receptor independent, nutritionally determined layer of AKT regulation that is active in cells and in a whole organism. And with that, I would like to thank my lab, Anna, Catherine, all of our collaborators, funding, et cetera. And we went over, but I would be happy to answer your questions.
Let's have a couple of questions if people have a few extra minutes. Yes.
I have a question. This actually is just like unlocked something that I've been struggling with.
Awesome. Okay.
Struggling with for a while.
Okay.
We have an E6 study that I think actually, I'm coming from Hopkins. People here probably did the metals analysis for me. And we found significantly depressed blood manganese levels in all the e-cig users versus the non-user controls. Whereas like manganese is a very common part of an e-cigarette coil, so we're confused why they would be having increased exposure to manganese but depressed blood levels. Which this could potentially be a pathway for why the manganese levels are depressed if it's activating this insulin pathway in their livers. What would be like long-term implications of this happening in your body and having continuously depressed manganese levels?
Depressed manganese levels, I can't tell you. We haven't done those, like that's not what we did. We did increase manganese levels. So that's what I can speak on. And also, I want to say that what we did was, specific to the liver. But what you pointed out is something that I'm like really interested in. Is there any connection between the ingestion or e-cigarette exposure to manganese and what's happening in the liver? Like that has not been established to my knowledge, because the manganese, there are some published reports that manganese can regulate its own transport. I would say they're not that strong. They're like, that's questionable. The transporter is definitely a regulated transport. We see very strong regulation by carbohydrates. Is it regulated by manganese itself? I'm not sure. But like, how can we connect exposure in different ways to what's happening in the tissue? I don't know. I'm not sure.
So cigarette, they have their livers, so we can actually.
There you go.
We just have one more question.
Two questions. Can you go over again how carbohydrates regulate manganese in the liver?
Yeah, it started to speed up at the end. So carbohydrates, this is a transcriptional effect. So, okay, this is well, let me start with this one. This is a fasting and refeeding time course. So all we did was we took away food from the mice for up to 24 hours, or we took away food for 24 hours and then gave them back food for, this would be like 1 hour, 2 hour, 4 hour, 6 hour, et cetera. And what you can see is that in liver, the expression of this efflux transporter is being induced by refeeding. And this diet is a chow diet, but it's a chow diet that contains 0.4% sucrose. It does contain sucrose, carbohydrate. Not all of them do, so I think this is important. We also repeated this on a high sucrose Western diet, and it shows the same effect. We did the same like fasting refeeding time course. Another group, we actually got scooped on this part of it, unfortunately. Unfortunate for Jen, but another group fed mice high fructose diet, and they also saw potent induction of SLC3810. So the carbohydrate signaling is occurring through this protein. It's a transcription factor, carbohydrate responsive element binding protein. And if you knock out this CHRBP from liver, specifically in liver, then you see a reduction in the expression of 3810, and now it can no longer be induced. Oh, some of my marks went away. It can no longer be induced by fructose. And then if we look functionally at the actual excretion, the egress of manganese from the liver into the bile, all we did here, we took mice, we gave them by gavage, we gave them like 100 microliters of water or 100 microliters of fructose, and then measured the biliary excretion of manganese. And that's what you're looking at here. So carbohydrate signaling requiring CHRABP is promoting manganese to travel from the hepatocytes out into the bile.
Very good.
The liver has the highest concentration of manganese in the body. So any excess manganese, is being transported to the liver. I don't know how that is happening, but I can tell you the transporter that's taking it up is called SLC 39A14. So that transporter is like scavenging the manganese from the blood to get it into the hepatocytes. And then from there, it's being excreted into the bile. What it's doing intracellularly is not known. Like, is it bound to something? Is it in a particular organelle? There have not been, it would be really cool and great once we have, manganese, what's the word I'm looking for? Like a tracer, a detector, anyway, like a fluorescent molecule that will fluoresce in response to manganese. And there are some like coming online, they're super expensive to produce, so we don't have it, but then we could start to understand like intracellularly, how is manganese getting across there? Does that answer your question?
Once we do the new blood we submitted to be able to do the spatial metal organ analysis.
That's true. That's true. Yeah. And I asked them about subcellular and they said it would just be a matter of like N number versus resolution. Yeah.
We have any questions online? No? Then I would say, let's finish here, but let's go to have coffee and drinks just the one floor above us.
Thank you.
And we can continue the conversation.
Thank you.
Thank you, Becky.
Thank you.
Which